No, I was just checking to make sure I was using the right
Yeah, I know.
sound source. Although, during the lockdown, I did have my little clapper in the laptop in my garage because I had audio recorded one way and video another way. And okay. Sorry this took so long. This has just been one long disaster of a day. We got good results this morning, but it was rough. So we're going to finally go through the exam. Gave you some pointers for the next one. And then, oh right, yes, I'm going to ask Annalise to send you instructions on hypocaudal measuring, because we'll need it for the other class as well. So we'll be on the lookout for an announcement on eLearn. Okay, and I'll try to remember to ask her to do that right now. So that you get it today. The e-notebook is down, but so you might not be able to do this for a while. I've got an open request. They're supposedly working on it. I don't know. Okay.
It's up again.
Is it? Oh, okay.
Yeah.
For now.
For now.
But luckily we don't need it. But actually, I will.
You're still down? Oh.
So Alice,
Oh.
I think it's just your last one.
No, it's down. It's definitely down. It's definitely down.
No, see, mine's not loaded.
Yeah, so it's down. It's down. Unfortunately. Oh, God.
Very, very, very down.
No, but
yes.
I'm out. Sorry about that. I'm out.
Did you pass it?
She's everything.
Tell me about it.
Picture with me.
Are you guessing?
Okay, now we're going to do it. So Annalise is going to do the thing and so. Okay. So overall,
Why do you stand there? Why are you just going that? You're not like this.
okay, well let's just go through the first one and then I'll talk about some strategies. Okay, and try not to talk too much because I don't know how well this is going to pick up my voice. So, I mean, answer questions and stuff, but just keep it down amongst yourselves. So the first one is, so GFP, gene or protein is transcribed to produce what? So this GFP gene is transcribed to produce mRNA, right? That's the only thing that's transcribed, which is then spliced to remove the introns. And I said in jellyfish. Why did I make that distinction? Where else did you use GFP? In bacteria, right? Do they splice? No, so that's why I wanted to make sure that we were back in a eukaryote. The GFP protein is translated from the mRNA by the ribosome. Now, some of you put rRNA, but it's really the whole thing, right? So I might have taken off a point if you just said rRNA. And then the GFP protein fluoresces when exposed to UV light. So basically this was kind of getting at the very first material, but also getting you to recognize when we're talking about a gene versus a protein, right? Now, some of you saw GFP and like, oh, transformation, and tried to use transformation words. So be very careful about that. Don't try to fit a pattern, right? Let the prompt guide you to the answers. Don't just seek, and then we'll come back to that in question 4, where you saw, oh, this sounds like experiment 1, so I'm just going to basically describe experiment 1. Well, it wasn't, so we'll come back to that, okay? So just make sure when you're answering these things that you're answering the question, not something that you're trying to fit that we did in class, okay? I'm kind of curious to see how you would have done if I used any other gene but GFP, but that's the first one that came to mind. Okay, any questions about that? So this is just really knowing your genetic information transfer terminology, right? And I didn't ask a single definition here, right? I put it in a context, and you needed to recognize and where they fit, Right? So that's the nature of these types of tests. Okay, so number 2, I gave you a table to interpret and ask you some questions. So we had LB was a lawn, LB plus can, 0, LB plus tet, 150, and then LB plus Hygaro, 0. And the prompt told you that kanamycin, tetracycline, and hygromycin were antibiotics, right? Just in case you had never heard those words, that's why it was there. Now, number 1, 2A says to discuss the observed growth pattern. So when it says discuss, it doesn't just mean describe, but it also means tell why, right? That's what discuss means, okay? So if you simply described the growth pattern, which is part of the answer, definitely, then you got part of the credit. But you had to say the why part. What's the why? Why do we see this pattern? What do you predict? Sure.
That the lawn has a resistance to, the LB has a resistance to TET.
Not the LB, what has resistance?
The bacteria.
Right.
Yeah.
And why? Why would they have resistance?
Because the resistance gene is activated by the agarose.
Not necessarily that activated, but the resistance gene was taken up when, yeah.
When they got transformed.
Right, with the plasmid, right. So the plasmid had the resistance gene to tetracycline and allowed it to grow. Exactly. It's just like experiment 3, except they changed the antibiotic on you. And almost everybody got that part. And maybe you didn't get the why, the part, the discuss part. I mean, next thing you can ask, you're like, is this all or do we want some more? Right? In fact, in one class, I just pretty much went and almost gave them the answer to number 7 because too many people were getting things wrong. But I didn't give them the answer, but I was like, maybe you should rethink that. Okay, so you can ask me. I'm pretty generous. What about, okay, so any questions about that? Right? So when you're asked to discuss, you want to go into detail. Not a lot, but just enough so that they know. That I know that you understand that the resistance is to tetracycline because of taking up a plasmid. Now, letter B. LB plus can plus tet. What about that? Would you expect the same bacteria from this to grow here? No, why not? Exactly. This is still inactive. This can still kill it. Right? So it only has resistance to this, so no. 0 growth. Exactly. And I think most of you got that, right? You gotta have a resistance gene specific to each of them as far as this experiment is concerned. Okay, now there are bacteria that are resistant to multiple antibiotics, but they have a gene for each one, right? They don't know that there's a broad spectrum resistance. Okay. In other words, one gene that produces a protein against a bunch of them. I think it's just that it's picked up many resistance genes. Okay? And those are the really nasty ones you get in hospitals, right? Misra, really bad. Okay. Any questions about the resistance? Yes?
So even though it has resistance towards TET, there will never be partial growth?
No, because this is dead. Right. Yeah, some of you did say that you might get some growth, but no. This tells you that can will kill everything. Yes. Good. Thanks for asking. Yeah. Okay. Yes. Yeah, okay. Okay, now we had 3 plates, same antibiotics, but through arabinose, lactose, and sucrose, and we see red here, coincidentally, right? Oh no, they're all TET. Never mind. So in this case, they're all TET. So we take these, plate them all on TET. Arabinose, sucrose, and lactose, and we see resistance on, I'm sorry, red fluorescence on lactose. So discuss. What did you come up with? Yeah. Exactly. So there's a conditional promoter with lactose activates somehow. Didn't ask for mechanism, but yes, then we produced a protein and we have red fluorescence. Okay? Good. So again, I wanted you to say conditional promoter, but again, it's similar to experiment 3, but the small molecule was different. Right? We did have arabinose, but it didn't work. And I think everybody understood that, but I really wanted you to say conditional promoter. Questions about that? Okay, excellent. Okay, so this one, number, oh, sorry, that's a different thing. Stop it. Okay. Eraser. So most of you got this, and it was good. And some of you did it a different way, and that's okay. I didn't even notice. So basically, so the prompt is, prednisone is a drug that alters gene expression. It's a corticosteroid. It's a small molecule that binds to transcription factor and activates gene expression. Draw a diagram of what's happening before and after. I didn't even read my own question. Where did prolactin go? Whatever. Okay. So the one I did was before. So we have our gene here. Here's our promoter, the circle. And I had my transcription factor blocking the promoter in before. And after, the prednisone, it's the black dot, the TF, can't bind anymore. And so polymerase can bind and transcribe. Okay, so in this case, the way I interpreted it was transcription factor blocks it, no transcription. Add the prednisone, the transcription factor comes off, freeing the promoter for RNA polymerase to bind and transcribe. Now some of you have inserted it a different way, and it's perfectly fine. So what's another way? Or the 3 students who took the exam late came up with it on their own. The other way is that, how did they do it? They did it where? They basically said that the promoter was there, but maybe there was something binding, but the promoter was there, but it was off. And then because in this case the transcription factor can't bind, but with the prednisone it actually recruited the polymerase and activated gene expression. Okay, so in my case it's repression, in this case it's activation. Both are perfect, answer the question. Okay, I don't remember. I was just grading on Saturday. I was like, wait a minute, that's not, oh, no, it is right. So, this type. So here we have activation, and here we have repression. Same thing, it works both ways. In fact, it probably does both in the same cell. In some cases, it may relieve repression. In some cases, it may actually activate. Okay. But that was it. I really actually wanted you to then go on to say, in this case, both cases where we got turned on, what happens next? So we got transcription. What's next?
Translation.
Translation, but what would we have
Splicing.
here? Splicing, and this would be what molecule?
Splices.
No. Pre-mRNA, right? So we get the pre-mRNA. But no one did this, so I didn't take off. And then you get splicing, etc. So basically a little diagram of genetic information. Transfer, right? And then translation. Right? Protein. So you can see on my nice little diagram, that's the way I answered my own question. But that's okay. I decided I'd take this. I'll have to be more clear next time to make sure I get everything. But this was really the important part. We already kind of covered this in question 1 anyway, right? And it's another thing, well, yeah, while I'm thinking about it. If you think you're answering the same thing for 2 questions, you might want to ask. Because I'm not going to grade the same thing twice, right? Unless I just write a really bad question. All right, I'm not going to ask usually that, okay? Any questions? Okay. And I'll post this for this class too. I'll post it for all my classes. In fact, I almost canceled class and just did it myself, and I was like, that's so boring. It's easier to have an audience. Okay. So one gene that prednisone turns on is the gene prolactin. It does, I checked. The scientist has exposed some cells to prednisone. So 4A is design an experiment. Okay, so here's something we probably haven't covered enough, so we're going to do it right now. So design an experiment doesn't mean to tell me how to set up PCR. Okay, and so if you did that, no points. Design the experiment tells me the, basically the write-up that you would get in front of experiment 1 or experiment 2. What is the, what tissues do I need, if that's the question, or what cell types, what DNA, RNA, protein am I going to extract, then how am I going to do it, and then how am I going to analyze it. Okay, so if we have cells, so here's our petri dish. Cells actually grow in a bottle. So here's our bottle of cells. It's liquid. And in this one we put prednisone. And we want to know what changes if prolactin turns on in this tissue culture. Okay? Maybe it's, I don't remember what organ produces prolactin. But anyway, we have some cells. We want to know if prednisone turned on prolactin. That's our question. So what else do we need? Is this enough? The 1 sample? No, I'll tell you it's not. What else do we need? A control, right? So our control cells, how would they be treated? What's that? No treatment, or the same buffer, but no prednisone, right? But so no treatment. Right. So we definitely need those 2 samples. Okay? And that would be sufficient for this question. Now what do, so we let that, we treat it with prednisone, no prednisone, we wait for several hours, days, I don't know, we wait some time. Time passes, now what are we going to do? We harvest these cells. What are we extracting? DNA, RNA. We're looking for prolactin expression. RNA, right? Expression means gene is turned on and we're making mRNA. Right, so that again, had to recognize what that term meant. So right, we're going to extract. So 1, treatment. 2, extract mRNA. 3. Okay, that's a good one. That wasn't in my, yeah, it was, I think. So we need primers. Exactly. Next. And just to be clear, primers for what? Prolactin, exactly. Right. So you needed to say that. So now what? Can we do PCR right now? We have mRNA. Can we do PCR? Exactly. We got to do reverse transcription. Right? What does reverse transcription give us?
cDNA.
cDNA. Now we can do PCR. And then what's next? Oh. I thought somebody said it. Gels. Yeah, just gel. Gel electrophoresis. Whatever. Wrong with gel. That's the design. Okay. So when we ask you to design an experiment, that's what I want. I don't want a master mix table or anything. Right. That's that. So we need to treat our cells. Well, treatment and control. Right. So you want to describe the comparison, right? Because we got to compare. And then extract RNA, mRNA, design some primers, reverse transcription of the mRNA for cDNA, PCR on cDNA, and gel. We don't need genomic DNA at all in this one. Okay, so again, let the prompt guide what you need. Don't try to fit it to what we did in class. Okay, just because in experiment 1 and 2 we had genomic, you don't always need it. Okay? Good. So then the gel. So in this case, based on this result, there's our wells, here's our ladder, whatever. So we're going to have no treatment and prednisone. So what would we expect in the no treatment? No bands, right? And here, a band. I didn't care where you put it. Right? That's not the important part. The important part is we would hope to see no treatment, and with prednisone, we see our prolactin. Okay? And that was full credit. But let's continue just because. What if you saw this? Wait, oh, what? We didn't add, we need something here. I didn't add something.
The water.
Yeah, exactly. So, negative control. This is also a negative control, so we'll call this the water. So this is clean, so our master mix is fine. We see a band here and a band here. What would that result mean? Yep. What's that?
You either messed up while preparing your cells and got prednisone instead of a treatment one, or prolactin expression isn't dependent on prednisone.
Exactly. So possibly a mistake, or this they both express is independent of, exactly. Right. So we'd have to find another cell type maybe, if we really want to study the regulation here. What was the other one I was going to ask you? So that's, well, it doesn't matter. I can't remember. Now if you saw this, now what? Screwed up step 5, right?
Yeah.
So we just repeat step 5 and try to be more careful, because we can't interpret this at all, right? Good. But that would be the result we would predict, right? If it's dependent upon prednisone. Okay. And this is in fact why, if you put on prednisone for more than 5 days, they have to taper you off the prednisone, because it does change gene expression. Okay. And throughout the whole body. So if you stop it suddenly, the body has problems. They put my cat on prednisone for a long time, and it took like 4 months to taper him down. And many trips to the vet for blood analysis. Okay. Questions? So this is what we mean when we say design an experiment. Okay. Great. Good, good, good. So for the next one, almost all of you got it. A couple of problems might have been in switching synonymous and non-synonymous. Just make sure you're using the right one. Right? Non-synonymous would mean what?
That it's
Amino acid is changed even though the codon, or sorry, right, the amino acid changed, right? So in the first one here is serine to arginine to arginine. So that was a SNP of a non-synonymous SNP. What about synonymous SNP?
same amino acid
Same amino acid, different
codon.
codon, right? And so here we've got alanines, but we got a GCT, a GCC, and a GCG. Because alanine actually gets all four. GCG, GCT, GCA, and GCEs, whatever, all four. Right? Indels you got, I didn't see a problem with that. Now, here's the thing, there were no TEs. Okay? So, if you put TE, I might have taken off a point, but yeah, there were no transposable elements in this thing. Now, the indels here, if you said yes or no and justified it, I gave you the points. Because in some cases, because the one indel is the two indels are deletions of 3 codons. Sorry, one codon. It deletes 1 codon, 3.
Base pairs.
Yes.
Yeah.
I'm
Well,
really.
bases.
Okay, I need to go and relax and calm down. Okay. Yes, a deletion of 3 base pairs or 1 codon may or may not affect gene expression. What I was thinking, I said in every case the indel is a multiple of 3, so probably no effect because there's no change in the reading frame. Okay. Some of you said no change for other reasons, or there might be a change because we were removing an amino acid or inserting. And that's, as long as you justified it, I gave you the points. Okay? Even though what I had in my head was different. But if you said it and it was correct, I have to give you the points. Okay? Good. Number 6 was I really just needed something to get points in, to get it to 100 points. But so what happens during the extension step of PCR? It's heated and extends the primer. And you had to be precise. I was looking for very precise use of terminology here. So PCR primers are extended to make a new strand. That's what I wrote, right? If you got anywhere like that, I gave you the points. What happens during the annealing step? Right, or bind or something like that, yeah. It's better not to, I did say anneal too, but saying that primers anneal during annealing is kind of not answering, you know. It's like saying a farmer or someone who farms. Right? That isn't really a definition of a farmer. But if you said that, it was fine, or bind, that was fine. The denaturation step, heat up and, right, to separate the hydrogen bonds, exactly. So that's, And the purpose of the primers, right, it directs the polymerase to the region, right? That's it. That's the thing. Okay? And then the order is...
denaturation, annealing, extension.
Exactly. And I purposely put them out of order above to make sure you read the question, right? Okay. And a couple of you did switch a couple of them, so just be careful, right? Read closely. Number 7. They were all false. I finally gave up in the end and decided that I would let you say SNPs or mutation. But otherwise, I wanted them all to be false. So, RNA polymerase synthesizes DNA. True or false? False, obviously. And it synthesizes what? RNA. Yes, RNA polymerase, RNA, mRNA, right? Yes. Transcription factor binded the promoter. It's false. Why? It bound the promoter. It bound to. Binded is not correct. Okay. We talked about it before. To bind becomes what? Bound. Just like to find becomes found. Okay. English is weird.
But your mind is not bound.
No. Yeah, that's true. That one is irregular of the irregulars. Yeah. Anyway, that was that. cDNA has introns. In this class, no. Okay. It had pure mRNA does, but in this class we've always said cDNA came from the mRNA. Right? It's not always true, but for this class, that's the case. P globe contains ampicillin. What's that? Somebody back here said it. The resistance gene, yes. The resistance gene to ampicillin. Ampicillin alone is what?
An antibiotic.
It's an antibiotic. It's a drug, right? It's a very complicated compound that does nothing about DNA. Okay. And so that also made you have to realize what is P globe. It's a plasmid, which is what type of compound molecule? DNA, right? So you had to recognize P globe was DNA. DNA has only DNA, no antibiotics in it. gDNA lacks introns. cDNA, or you could change it to genomic DNA has introns, right? I would have taken either, but if gDNA lacked introns, we wouldn't be talking about splicing. Reverse transcriptase synthesizes mRNA. No, what does it synthesize? cDNA, right? Yep. SNPs are a type of mutation. I was really looking for polymorphism. It's not really considered a mutation unless we know the wild type or the ancestral, right? And even so, even if we know the ancestral is one amino acid, it doesn't mean that the different one is a mutation, right? It may just simply be a new variety. So really, when we're just talking about a SNP, it's a polymorphism. And like in my morning class, we made mutations, right? We designed things to mutate the gene. So mutation, yeah. So try not to use that word too much. mRNA is double-stranded. Single. Primers cut DNA. Yeah, you could say helicase cuts to DNA, or primers blank DNA. Bind to DNA, right. rRNA carry amino acids. tRNA, exactly. Okay. And I think almost everybody got the PCR table. Okay, finally. There were a couple of, on the PCR table, there were a couple of times where you used a different subtotal. Just remember, it's always total volume when you are calculating one reaction. Hold on just a second, because I want to talk about some strategies. So any questions about the material of the exam? We finally did it. 4 weeks late, but okay. Our project just didn't work out so well to let us do this. The project worked out great. Don't get me wrong, the results are really good, and Annalise is very excited about them. So when you get a test like this, I suggest you read every question. Take a few minutes, take a deep breath, and read every question. Answer the ones you think you can get through with pretty quickly, right? And then go back and tackle the ones, like maybe the design and experiment. But they say, I don't know if it's true, but they say that if you read them all, your brain can actually start kind of churning on all of them, right, in the background while you're focused on one. And so it might help trigger some things. Like I said, don't try to fit something into one of the 4 experiments that we did. All right? We're going to draw from all those experiments to get you to design new things, right? We want you to mix and match now. Basically what the 4 experiments give you is the toolbox, right, that you can use. And we didn't even cover sequencing and splicing in this case, or sequencing and blast here. Okay. So, right, but those are the tools that we've given you. Now we want you to recognize when you need to use each tool, when you're given a new scenario. Okay? Because that's what scientists do. We come up with a question, then we go look for the tools, right? So that's what we do. If you find yourself telling me to label tubes and things like that, take a minute, erase that, and start over again. Okay? I don't ever ask those questions. Ask for that. It takes me long enough to grade this stuff. I want content, good content, right? There will be some stuff from Annalise's project. We won't get too heavy into the epistasis. We ran out of time to really help you really understand that logic. I mean, you were starting to get it the other day, and that was great. But I think you're getting it more than you thought. But it's not easy, right? But it is really cool. And if you take some time to go through it yourself, I think you'll find this really cool. And that's genetics. That's the stuff I loved. One of my favorite class of all time was my graduate class in developmental genetics. And I mean, we learned all that stuff. It was really fun. I think I was the only student in the class who thought that, but that's okay. But the overall goal of a project, how we tackled it, that we definitely want you to know. But again, there's more tools there that we use to answer her question, or address the question of what is this plant, right? That was pretty much your question. If you don't understand a word in a prompt, just ask me. I should be able to give you a definition or find another word to help you. Okay. And sometimes maybe the whole class needs that. I grew up on a different side of the country, so I oftentimes use words that aren't as common here, and vice versa. Right? So I'm trying to think anything else about the exam. Taking strategies. Then, just to wrap up a few things, because we won't be meeting on Wednesday. I'm only going to grade the faculty assignment that you've turned in. We're not going to do a second draft, as said. I just can't. I don't have time. Okay, I got to write a midterm. I got to look at your intros and grade midterm and whatnot. So we're not going to do the second round. I'll just grade the first one. That's the full 30-something points. Whatever it is. We're not going to complete the report. The intro will be graded because you turned that in, but you won't be completing the rest of the report. You will, on Monday, a week from today, you will be doing your presentation. Okay? I'm going to be posting the guidelines. It's really simple. The guidelines will tell you you have 5 minutes, and most of the time the students don't take the 5 minutes. You get like 6 slides. and the guideline tells you what goes on each slide, okay? It's very, very specific. So as long as you've got that and you can talk about it coherently, you'll do really well. Okay. Try not to be nervous, although giving a talk in front of your peers is actually the hardest to do. Please tell me it's still recording. No, I think it is. Okay. Yes.
Do you know how much of Annalise's project will be on the final? What percentage?
No, but it won't be like the whole thing. It'll be
half.
not like you're thinking.
Okay.
Yeah.
Are you gonna go over it during?
Yeah, I'm gonna go over it.
Yes.
I'm actually gonna explain it to her,
Okay.
just so she knows what I'm saying.
Okay.
Right. And then he is going to cover it, so make sure you go to his review. Before the presentation, just a few things. You can wear just like what clothes you're wearing today. You don't need to dress up unless it will make you feel a little more confident. But please don't show up in a three-piece suit. I've had students do that, and I think they're actually more nervous now because they just felt out of place. Do practice, okay? You'll feel more confident. When you do practice, so in biology, we don't give talks that are written out, right? We don't script it and stand up there and read it. The slide is our guide outline, and we look at the slides and we talk, okay? If you want to have some note cards, that's okay because you're learning, okay? But that's just not what we do. The talk itself is sort of extemporaneous, but the slides are what guide us, okay? But you do practice. It's a good idea, something I learned on my own way back when I started giving talks, is to memorize the first two sentences though. That gives you a chance to start talking in front of a group of people, hear yourself in a room, and then go into the unscripted part. And I still often do that when I actually give talks. But my first talk, it was horrible. I crashed and burned, and so I actually made myself give as many talks as I could, volunteer. So now I don't care. I can talk in front of anybody. So right, what else? Oh, on your slides, make sure they're good contrast. My slides are usually pretty boring, black on white. Okay, you can do yellow on black. Blue on black is ridiculously hard to see. Okay, red on black, very hard to see. The contrast on your computer screen is probably you're going to be able to see it, but the contrast on that thing you're going to lose it. Okay. Also, at least 10% of the audience is going to be blue-black colorblind. Okay, blue, yeah, and so they're going to see a gray screen, right? That's it. So nothing. Okay, 3 or 5% will be red-green colorblind, but we won't worry about that. No GIFs, GIFs, however you say. I will fail you. No, I'm just kidding. Don't use them, please. They annoyed the crap out of me. They're so distracting, nobody's listening to you talk. Okay. If you need a video, make sure it can loop once when you press video. Go, right, and it stops. Okay. What else? You can, if you have empty space, so pretend this is a slide. This is the most important part of a slide because we read right to left, top to bottom. This is where the good stuff goes. If you've got empty space down here, a picture of a Arabidopsis, something to fill the space is nice. A gratuitous image. No gifts. But, you know, if you don't have something relevant, you know, you can always throw in a picture of a nice flower of the Arabidopsis. Something relevant, but you can fill in the space. But it should be in this area. This is where the important stuff goes in a slide. I mean, look at billboards. See how they're set up. They're either here, you're here, right? The boring stuff is down here. Again, your slide should be your outline. So, you know, if you need to, take 10 seconds, look at your slide, read it, and then turn around and talk. But don't talk to the slide, talk to out to the audience. Other than that, don't worry about eye contact, all that stuff they tell you when they're trying to learn to do talks. I'm not looking for that. I'm listening to what you're saying and I'm looking at your slides. Okay, that's it. Yes?
Will there be a posted grading rubric?
Not necessarily a rubric, but the slide the handout will guide you completely through. So we will have a rubric for grading, but I don't usually post it because it is pretty, so only I can understand what it means. Usually we have 3 people review them, grade them. I don't know about next time, and probably maybe just me, and I do ask the ULAs to fill out the rubric, but I don't actually deal with the final score. Only I do that. It just helps average out the stuff. But I'm really generous. Okay, so this is, but do take it seriously. Since we're not going to finish the report, this will be the only way that you sit down and really think through the project, get it in your mind logically. Talking about something is very different than just stare at it on a piece of paper, right? Writing about it is very different than talking about it, which is why I wish we could do it, but it's not going to happen. Yeah. So go to Ibrahim's review. He is going to review the project, which is very nice. I'm not comfortable doing it because I have 3 projects, and I'm not an expert in any of them right now. I haven't had the time. I'm the technical. I'm down here, you guys are up here, and that's what Ibrahim will help you with. I've had so much in this area that I really haven't taken the time to learn the projects, so I would be afraid of giving you the wrong advice, right, telling you wrong. I keep saying we'll never do 3 or 2 or more than 2 projects a quarter, and it always happens. So it is rough, let me tell you. And we lost our person who usually took care of all this part for us. And not lost her, she moved. She's still she moved to another job, which we know and we were expecting, and I'm glad she was able to. But I really miss her because it's been rough. You never realize how much people help you out until, I mean, I knew how much, but not really, really, really how much. Okay. Any questions? So Monday, Ibrahim will be here and talk. We're going to listen to your talks, but he won't come on Wednesday because we don't ask them to proctor. We will be sending out we will be sending out an evaluation for Ibrahim. It's just to give nice feedback and ask you to fill it out. But Monday will be the last day he'll be here in class. Then maybe Monday, depending on how long the stuff takes, we can talk about the outreach group and becoming a ULA in future if you choose to do that. Annalise will come for the talks on Monday too, and probably won't come on Wednesday. I'll just sit here and proctor the exam with you guys. My second least favorite thing to do with my job. Rating and grading the exam is first. But we got to do it. We got to do it. Okay. To be honest, there are 2 reasons why we do the exams, in my opinion. One, it's the only time that I know it's you and the material, right, and not you and a neighbor and you and the internet. And I know you're not cheating or whatnot, but still, it's the only time I know what's in your head. Secondly, it's really what gives me the A plus B, the plus minus break, because you have so many points. This is what really distinguishes you, is the 2 midterms I have found, because we used not to do them, and we had to add them in, and that's what I noticed, is that it helped us with those breaks. Otherwise, once you leave college, in grad school, whatever, get out in the real world, you'll never be given a piece of paper and told to solve a problem, right? You can always walk down the hallway and ask somebody. This exam thing is so unique to college, right? But in the real world, it's collaborative, right? So anyway, you can study together, work together, but on Wednesday we'll do that. And you can practice with the talks together if you want. I don't care. That's good, right? Practice with each other and get some feedback. But okay. Did you have something to add?
Are you also going to talk about RISE?
Let's do that next time. Yeah. I'll do it individually to each class. Yep. And also, may not. I'm sure RISE will happen, but there's a big problem right now. But that's not your problem. something you guys don't need to know about. So I don't know how much I need to advertise right now. And I won't see Connie on Wednesday. Anyway, any questions, email me, I'm available. I will see, I guess, back here on Monday. Have a good break, eat lots of tasty food. And that's that.
Thank you.
Thank you.